Hello, my name is Aaliyah, Cano, and today we'll be presenting my research on characterizing supermyces, services, and 9448 and generating ethanol tolerance. Now, those of you who have experienced every winter storm / freeze in Austin since 2021 might agree when I say that climate change is a real and growing concern. So that change in temperature and weather patterns we're experiencing is attributed to a buildup of greenhouse gases such as carbon dioxide in our atmosphere. Transportation accounted for the largest proportion of greenhouse gas emissions in the United States, and this is because we rely heavily on a fossil fuel called petroleum to make the gasoline that powers our cars. So understanding the negative impacts that petroleum and fossil fuels have on our environment has prompted many to search for fossil fuel alternatives, and one alternative of interest to us is biofuels. So in contrast to fossil fuels, in which fossil fuel drilling, processing, shipping, and burning release carbon dioxide at every step, biofuels have an almost net zero contribution of carbon dioxide to our atmosphere. And to best understand why, we have to start with the plants. So the plants that we farm are shipped to a fermentation processing center where they're used to produce ethanol. And the ethanol that's produced there replaces the petroleum found in gasoline, so that when we drive our cars, we end up burning ethanol rather than burning petroleum. But this is not to say that when we burn ethanol, we don't release carbon dioxide into the atmosphere, because we do. But instead, the carbon dioxide that's released is used to grow the next generation of plants that will then end the cycle. Hence that net zero contribution I was talking about. But I kind of skipped over this step, and that's because this fermentation step is a bit more complicated than I'm making it seem. So fermentation is carried out by microorganisms such as yeast, who are in need of making energy molecules called ATP to survive, but they don't have the oxygen available to them to do so. So basically, microorganisms like yeast will carry out fermentation, but at some point they'll also end up making ethanol as a waste product, which is harmful for them, but it's beneficial for us because we can use it. Now, there are different plant biomass types that we can use in this fermentation process. The one that's most commonly used today is corn, or first generation biomass, and that's because you can figure it's fairly simple. But if we want to see a greater impact on our climate, we have to use additional plant biomass types. And this includes second generation biomass, and more specifically, my lab and I are interested in fermenting pectin-rich biomass. So this includes apple pomace, citrus waste, and sugar cane. The reason why we aren't already fermenting pectin-rich biomass is because, as you can see in this figure, the cell wall structure for pectin-rich biomass is a lot more complex than corn, for example. So this means fermenting pectin-rich biomass is a bit more difficult. So if we want to begin fermenting pectin-rich biomass, we would need to, so we'll compare the two so you can see what I mean, why it's a lot more difficult to ferment pectin-rich biomass. So if we want to ferment corn, for example, we would have to add an enzyme that breaks up the polysaccharide called starch, which makes up the majority of the plant cell wall structure of corn, just so that way we can get the individual monosaccharides that we can then feed our yeast and produce ethanol. But if we were to repeat this process using pectin-rich biomass, we would have to add a lot more enzymes to our fermentation process, just so that we can get the individual sugars that we would then feed our yeast and end up producing ethanol. And these additional enzymes are not cheap. Fortunately, a yeast strain called Saccharomyces cerevisiae 09448 was found to produce its own pectinase enzyme, which makes fermenting pectin-rich biomass a bit more affordable. But it's unclear, however, if 09448 can withstand the industrial conditions they would be exposed to if they were to ferment pectin-rich biomass on a large scale. So basically what I'm saying is we don't know if it can withstand increased temperatures, increased osmotic pressures, increased acidic conditions, or increased ethanol concentrations. And if it turns out that 09448 cannot withstand these conditions, there's hope that we can use a mutant mutagenesis protocol to help us produce a mutant that can. So this brings us to our first research question of the day, which is characterizing Saccharomyces cerevisiae 09448 under industrial stress. So before we jump into the actual procedure for this experiment, we first need to identify a positive control. We needed something to compare 09448 to. And the positive control we chose was an industrial strain called active dry yeast. And so as I mentioned, active dry yeast is an industrial strain, so it's already used to those industrial conditions. So now the question becomes, how does 09448 compare to active dry yeast? Does it go better, just as well, or worse? So now it's the actual experimental procedure. We exposed active dry yeast and 09448 to different strengths of these conditions and measured their growth using a plate reader for 16 hours. And more specifically, we exposed active dry yeast and 09448 to 0% and 20% ethanol concentrations. For this experiment, we induced osmotic stress by using different salt concentrations. So a low salt concentration is equivalent to a low osmotic pressure, whereas a high salt concentration is equivalent to a high osmotic pressure. we exposed them to 0.09 molar sodium chloride concentrations and 2.0 molar sodium chloride concentrations, and everything in between. We exposed them to pHs ranging from 3 to 6, as well as temperatures ranging from 30 to 43 degrees Fahrenheit, and again, measured their growth in these conditions using a plate meter for 16 hours. So before we go into the results for this experiment, we need to understand what a minimum generation time is. And a minimum generation time is the shortest amount of time it takes for a cell population to double in size. So say we have a blue cell population, for example, and this cell population takes 60 minutes for it to double in size. And so we also have a red cell population, but instead it takes 120 minutes for that population to double in size. We would say that the blue cell population has a shorter minimum generation time, and therefore it's reflective of it growing better in that environment because it's growing more quickly than our red cell population, for example. So now to the actual results, we found that as temperature increased and as sodium chloride increased, 0.9448, which is found in blue in these figures, experienced increased difficulty growing. We found a similar pattern with ethanol concentrations, in which as ethanol concentration increased, 0.9448 experienced increased difficulty growing. But fortunately for us, as the environment became more acidic, so reading from right to left here, as the environment became more acidic, 0.9448 grew relatively consistently. So it's fortunate for us because this is like one of the conditions we don't have to worry about too much. But overall, we see that active dry yeast has shorter minimum generation times in most of the conditions we expose them to. So this basically tells us that active dry yeast outperforms 0.9448 in most of these conditions, and that 0.9448 is not yet ready to be used industrially. This brings us to our second research question, which is generating ethanol-tolerant mutants using a mutagenesis protocol. And I like to think of our mutagenesis protocol as maybe little yeast/Hulk cells, and this is because similar to how gamma radiation was used to help turn Bruce Banner into the Hulk, we use UV radiation to induce random DNA damage in our cells, and this allows us to produce mutants. So now the question becomes, which of the mutants we produce is better at growing in high ethanol concentrations than our unmutated version? So once we exposed 0.9448 to UV radiations, we had to identify which of the mutants we produced were showing some initial signs of ethanol tolerance. And we identified those mutants by growing them on alveolar plates containing 10% ethanol. So whichever mutants were growing on these ethanol plates, we organized them onto a patch plate, and then from that patch plate, we chose 12 of them at random to further characterize. So by that, I mean we chose 12 mutants and we measured their growth in 0%, 2%, 4%, and 6% ethanol concentrations, and compared their growth to an unmutated version under the similar conditions. And overall, we measured all of those organisms' growth using a plate meter for 36 hours. So I know we just learned about what minimum generation time is, but it's also important that we understand what final optimal density is. So optimal density is a way that we measure the concentration of cells in an environment. So say, for example, we measure a cell population at hour 1 and at hour 25. We see that at hour 1, the cell concentration is really low, or fairly low, so we would say that at that time, that cell population has low optimal density, whereas at hour 25, we see the concentration is higher, so at that time, we would say that the cell population has a higher optimal density. But more specifically in this research, we're interested in final optimal density, which for us was the greatest concentration that the cell populations were able to grow to, and this is reflective of their ability to grow in that environment. So say, for example, we have two strains, strain A and strain B, and we see that strain A reached a higher final optimal density than strain B. We would say that strain A is growing better in that environment because it grew to a greater concentration than strain B. So now to the results of this experiment, we found that over all 12 mutants that we examined, mutants 1, 2, 4, 6, 7, 9, 10, 11, and 12 reached a greater final optimal density than our unmutated 0.9448 strains, so they all grew over those red bars there. We also found that mutants 1, 2, 4, 6, 7, 9, 11, 12 also had shorter minimum generation time, so they grew more quickly in those environments. So overall, we found that mutants 1, 2, 4, 6, 7, 9, 11, 12 were our 8 mutants of interest because not only did they grow to a greater concentration than our unmutated version, but they also grew more quickly when exposed to 4% and 6% ethanol concentrations. And these figures are pretty, like the colors, you know that is great. But the only thing that's missing from these figures are the error bars. And the reason why we don't have error bars is because we only conducted this experiment once, and this was more to kind of funnel out which of the mutants weren't worth re-examining again. So now that we have our 8 mutants of interest, we repeat these experiments again, but only testing them under 0% and 6% ethanol concentrations. And so when we did that, we found that In contrast to what we had seen before, only mutants 6 and 12 were able to grow to a greater final optimal density than our unmutated version, and those two also had shorter minimum generation times than our unmutated version. So we put those two data sets together, we see that mutants 6 and 12 are our new two mutants of interest because they grew to a greater concentration and also more quickly than our unmutated version at a 6% ethanol concentration. When we plot their growth curves, however, we see that the error bars in this figure are huge. And this basically indicates to us that there are some inconsistent growth patterns within these individuals. So every time we repeat these experiments, so that's why there's three different colors here. So it's blue, orange, and gray for the first time, and the third time we repeat these experiments. We see that their growing pattern wasn't the same every time we did them. And in fact, mutant 12 had more consistent growing patterns than mutant 6. So this means that overall mutant 12 is our most promising mutant because it not only grew to a greater final optimal density, but it also grew more quickly and grew more consistently in higher ethanol concentration than our unmutated 0944H strain. So in the future, we want to examine 09448 under higher ethanol concentrations as well as the other industrial conditions that we didn't examine them under for this experiment. So increased osmotic pressures, increased temperatures, and increased acidic conditions. Additionally, as I mentioned in our mutagenesis protocol, we induced random DNA damage to produce these mutants. So in the future, we want to sequence the DNA of mutant 12 and see exactly which genes did we break that allow this mutant to withstand these conditions. But if it turns out that mutant 12 is capable of withstanding these industrial stressors, there's hope that we can use it to ferment pectin-rich biomass on a large scale and produce more biofuel that will overall lower the impact that transportation has on our climate. I would like to thank Dr. Edwards for her mentorship and her guidance as I conducted these experiments. And I wouldn't be the microbiologist I am today if she wasn't such a great mentor, so I'm so grateful for you. I'd also like to thank Jennifer Foster for the meeting, Jordan Macario, Bryce Jones, and Sophia Martin for helping me collect the data that I presented here today, as well as Bashir Sager and Derek Cardenas for their support in the lab and attending to our meetings when me and Jennifer couldn't do it, as well as St. Edward's University, the Department of Biological Sciences, the Gutierrez program, and I for funding it. Thank you. Thank you. Let's start. We got there.
Not right, sorry. I'm going to start by having an advisor, Dr. Edwards, come up. Dr. Edwards is going to say a few words about working with Aaliyah and also present the cord and stole that I hope you wear with pride and graduation to represent this and everything you've done in honor before you head off to get your PhD. Congratulations.
Well, thank you all for staying so late to see Aaliyah present. I know it was worth it. Aaliyah started working with me her first summer as a first-year student here, and I was really lucky to have her come join me in the lab. She took one summer off, which broke my heart, and studied fish. Yes. At night. At night.
I don't know if I felt about that. That's why I can go.
Before she decided to come back, and I was so excited to have her because Aaliyah just has an excitement for science that I don't think I've ever seen, and it's contagious. And we're working on a manuscript now, and the fact that she's excited every time she sees me about like going in little tiny details and correcting like things that the minutia that drive most people crazy like helps me make it through every week. So thank you so much, Aaliyah. I'm so happy.
Questions for Aaliyah?
Any questions about the beginning stages of your UV mutation process?
Yes.
So I saw on the slide when you were talking about the plates where you said 10% alcohol. There was also a plate that said you had a certain percentage of glucose, so the concentration.
Yes.
Is that something that is a future experiment for this lab to create more glucose resistant use, or that's
So this plate, the reason why we took this here is because we needed to see if our ethanol plates were actually like strong enough to kill off some of the non-resistant mutants. So basically we saw, like, so you can see in this other set of plates, you see how there's more mutants on the yellow plate? Yeah. So that's our control plate. So that basically tells us that our ethanol plate is working in kind of eliminating the non-ethanol resistant mutants. But osmotic, or I know of you were working with us when we were doing the sugar experiments. And so there is, like, we found that there's only certain sugars that 0.9448 could use in the fermentation process. And we also know that when we increase that's what the osmotic pressure experiments are, is like increasing concentrations of just stuff in their environment. So, yeah, that is like a concern, that if we have too much sugar in that environment, like how would it react to that increased osmotic pressure?
What could be the reason behind the inconsistent growth patterns of the insect shell and graduate before?
Yeah, that's a good question. It could be. So and I know we don't like to say this. My bad. But it could be like the way that we set up the experiment. So the thing is, whenever we set up the place, we have to be so tedious, and we don't know if we're always inoculating the same like stuff in each well that it doesn't come in. So it's possible that there's just like something slightly off with the environment that we're being used to grow in. But again, these are also like random DNA images in these cells, so it's possible like the organisms themselves are like slightly different from one another. Because like we can't guarantee that all of those yeast are identical, but most of them are. Is that take a look. Okay. Is that okay? I don't know. Yeah. Because like that's why we choose like individual columns, because everything in there should be like identical to each other, but like you can't always guarantee that. Like we should. But that's a good question. I don't know.
So like when you inoculate to start these, do you know you're starting with the same number of cells?
Yes, so we make sure that we're like get the. I'm trying to go back to the actual procedure. Because like we have to find the original like optical density.
You start
to
with the same
off with.
OD.
Yes, yeah, and so we do the math to make sure that we start off with the exact same numbers. Well, not the exact same. That's as close as possible. Yes. So.
And do you, when you are thinking about adding on additional stressors, are you going to start with your ethanol tolerant mutant and then try to mutate that, or do you go back to the beginning? Like how are you gonna? Because ideally, in the end, you'll have a mutant that's ethanol, acid, osmotic stress, temperature resistant, right? So do you do one at a time and just keep adding on, or do you go back to the beginning and somehow then make double, triple mutants?
That's actually a good question. Because like we have like the mutants that we have right now that we tested their ethanol tolerance, like we have them on plate. The original mutant 12 right now that we have is in the cryostop. So we can't like it's hard to we're constantly like replating them and reshaping them, so I don't know how they're changing over time compared to our like cryostop that we have. So I'm thinking that we maybe start from the cryostop again and like re-examine them under these just different industrial.
But start with that mutant.
Yes, yeah, and so yes. But that's so I was pretty.
I think that's what, if you want to finish up answering that question, yeah, please finish your thought, and then we'll move on to our last.
Okay. So whenever I presented this research at Southern as a poster presentation, it was brought to my attention that like we have to measure their resistance to all of these stressors at one time, like rather than just like individual stressors at once. So like that's also something that we need to do in the future. Like we know that they're resistant to these things when that's the only stressor they're exposed to, but what happens whenever they're all exposed at the exact same time? That I know we don't